Hi, my name is Suzan Ok, and for my MPhil in genetics, I am investigating the invasive potential of a defensive symbiont. And our ultimate goal is to use an optimal strain of a bacterial symbiont to quell viral disease transmission by mosquitoes. So arthropod-borne disease is a big issue all over the world. For example, Aedes aegypti is the primary vector for dengue virus, which is very serious in many tropical regions. So basically, we're looking at a defensive symbiont, and it's an endosymbiont. So what that means is it's heritable. So it's located within the insect, and it's passed on to subsequent generations. Symbionts can have parasitic effects, such as manipulating host reproduction in order to favor its spread, and they can have mutualistic effects. So these are benefits to the host, such as protection against pathogens. And I'm working with Wolbachia, which is a very common bacterial endosymbiont, and it has both parasitic and mutualistic effects. It's intracellular, so it resides within the cells of insects, and it's transmitted by females through the egg cells. And it's actually found in up to 40% of arthropod species, so insects such as the fruit fly, even crustaceans or arachnids, and it's also found in filarial nematodes. So one of the effects of Wolbachia is antiviral protection. Basically, this has been measured by looking for a decrease in viral titer for a wide range of RNA viruses. Basically, insects that have Wolbachia and are challenged with virus, such as Drosophila C virus or flockhouse virus, will show a decrease in viral titer over time. It's also been shown to protect against more serious human RNA viruses like dengue, chikungunya, and West Nile virus. And one other thing is that there's between-strain variation, so not all strains of this bacterium will confer antiviral protection, and we're interested in finding one that has a high amount. Wolbachia also has this phenomenon called Cytoplasmic Incompatibility, or CI. Basically, this is a model for it. So if you have 4 different types of crosses, uninfected females crossed with uninfected males, so they don't have the bacterium at all, they'll produce viable offspring. But an infected male with an uninfected female may have CI. So that means that during spermatogenesis, some sort of chromosomal aberration occurs and the offspring are not viable. So the eggs that the flies lay don't hatch. The reason cytoplasmic incompatibility is so interesting is that infected females will produce viable offspring with any male, whether the male has the bacterium or not. So according to this model, an infected female has a reproductive advantage over an uninfected female. And that allows the bacterium to maintain itself in the population of insects, or of hosts. So with antiviral protection and CI, basically Wolbachia can achieve a high enough frequency in the population and kind of reach this threshold where it is present in the population over a long period of time, which is interesting for us because we want it ultimately to decrease the amount of virus in the host so that the host can't transmit viruses. So I did a variety of experiments this year, and one of the ones that had the most interesting results was on CI. So basically, we did all 4 different types of crosses, and we looked at how many eggs the female flies laid after the crosses and counted unhatched eggs to determine the egg hatch rate. And what we found is that, and this was with 16 different strains of this bacterium. So some of the strains didn't really show any CI. They had a very high, egg hatch rate. So those are the ones on the right. But a lot of the strains on the left had 50% or fewer of an egg hatch rate. And that's really interesting because this is the kind of thing we're looking for. They're basically, when you have an infected male with an uninfected female, you're getting CI, and then infected females are advantaged. So basically, that's what we've been doing. And I think I lost a slide. But another thing I'm doing right now is looking at 14 different species of Drosophila and basically studying antiviral protection in them. So looking at the species that have their original bacteria inside. So we're interested in basically understanding how common it is in different species of Drosophila and whether we could use one of those strains to prevent disease transmission. So I'd like to acknowledge Frank Jiggins, my supervisor, and Julian Martinez, who has been mentoring me throughout the year. And then also the Department of Genetics, the University of Cambridge, Churchill College, and the World Bank for my funding. Thank you. Yes?
Is there any connection between CI and viral protection? Do they tend to occur in the same strains?
That's ultimately what we want to find out. So we still have to compare that basically, but we're looking for something that has both CI and antiviral protection. So there have been some previous studies on this, and we're sort of, so actually they have introduced strains from Drosophila, from melanogaster into mosquito populations and found successful results, but we're constantly trying to find more strains that could work. Yes?
Is it known the mechanism by which Wolbachia reduces viral load, and is there anything known about that in an evolutionary context?
So certainly protecting the host long enough for the host to transmit the bacterium to the next generation is good for the bacterium. In terms of the mechanism, it hasn't really been elucidated, but so Drosophila has a very complex multi-layered immune system, and they think that the presence of the bacterium within the cell may actually interfere with the replication of the virus, because the virus, if the bacterium is in the cell, the virus is in the cell at the same time, maybe there's some interference occurring there, or the bacterium may basically be upregulating immune pathways. So there's IMD and toll pathways that the fruit fly uses, so maybe it might be a combination of those things. Yes?
Is it a big problem with when you choose an insect like Drosophila, you want to be sure it's got just this one bacterium that's relevant? It's loaded with all kinds of other things.
Certainly. I mean, that's true. I guess that could be one of the obstacles to this research, but generally, so for example, with what we did here, we took the 16 strains of the bacterium and put them into one species of Drosophila with a very similar genetic host background, so we would think that at least we've kind of diminished confounding factors there. Any other questions?
Confusion in my head, which I certainly haven't understood. Normally, if I think of something that's being affected, it is at a disadvantage,
Yes.
etc. And you're using the word advantage.
Yes.
Right? So
It's a bit confusing.
That
Yes, so it's a bit complicated because on the one hand, it can have costs on the host as an endosymbiont because it's using up resources, but if the benefits of antiviral protection outweigh the costs, the fitness costs it might have, so yeah, the host is infected with this bacterium, but it's sort of a symbiont, so it basically, it's kind of co-evolved with the host at that point. Exactly. Any other questions?